This is a production of Cornell University.
Hi, everyone. I'm really happy to be here this afternoon and share a little bit of what I've been doing at Cornell for the past year. And so Don just gave an introduction to what I'm doing here. I'm an anthropologist by training, and I'm taking advantage of this opportunity that the NSF offers for social scientists to gain experience in fields outside of their major field. And so whenever I talk about the lost crops of Eastern North America, it requires a lot of exposition because most people have never heard of them. So I'm going to kind of start at the beginning and give you a little brief introduction to how archaeologists convinced ourselves and others that these are indeed lost crops, that some of them were domesticated. And then take you through some aspects of my dissertation research that led me to the questions that I have about the role of plasticity in domestication, which is what I'm exploring here. And at the end, I'll show you some of the very kind of preliminary observations or some of the things that I've been doing here. Our plants are still in the field right now, so we'll be harvesting them soon, and then we'll have more results from this year. So first of all, my background. What I've been trained in is Paleoethnobotany, which is a subdiscipline of archaeology. Paleo obviously stands for old. I analyze ancient plant remains that are recovered from archaeological sites. Usually, in my case, seeds and fruits, but other small fragments of plants will do. Ethno is people, so ethnobotanists talk to people a lot. I sometimes talk to people, but because I study ancient agriculture, I more often will read historical and ethnographic records to understand how early, agriculture worked in the past. And then botany, I don't think really needs an introduction in this crowd. So just to kind of give you an intro to how we get the types of plant remains that I analyze or that I specialize in, it's extremely simple. It's a method called flotation. So basically when we find contexts where plant remains are likely to be found, such as this hearth, which the flotation sample has already been removed from, this floor that has tiny pieces of charcoal embedded in it, or this area around a grindstone. Those seem like good places to find plant remains. We take a soil sample and we process it by flotation, which basically just means that we pour it into a bucket or a bin of water, agitate it, and then pour off the organic material that floats to the top. So that's what my students are doing here. They're skimming the organic material into a cheesecloth, which we then dry out. It's very high tech. And we bring it back to the lab to analyze it. So hopefully once we've picked out all of the sort of roots and modern seeds and things from the sample that are not of interest to us, what we're left with is ancient plant remains that were left behind by people. And because I study domestication, I usually take this one step further and take microphotographs or some sort of images of these seeds and fruits and then analyze them to see if there's been any change over time in the size and shape of the fruits or if there's any differences geographically. So this happens to be the species that I studied for my dissertation. Just to give you an idea of kind of what my data looks like. There's a lot of variability, obviously, but there's also variability in how well preserved these are. These are all exceptionally well preserved, which is why I bothered photographing them in the first place. But you can see that they're not perfect. And so another big aspect of what I do is kind of trying to reconstruct what they look like before they were burned and buried in the ground for 2,000 years. But I'm not really going to talk much about how we do that today because it's actually very boring. So domestication is what I specialize in, and the way that I define that is a coevolutionary relationship where human practices are changing the bodies and behaviors of plants and animals. And of course, if it's a coevolutionary relationship, I strongly believe that our relationships with these plants and animals are also changing our bodies and behaviors over the course of thousands of years. So the reason why I chose these two images to represent domestication is because these were two very simple experimental projects that taught us very important lessons about how domestication works. So in the case of Baliev's fox experiments, what he did was he just selected wild foxes on one specific trait, which was lack of aggression towards people. And what ended up happening over the course of several generations is that a whole suite of other traits that our domesticated mammals share, things like shorter birth intervals, piebald coats, floppy ears, all these different things that many of our domesticated animals have, ended up kind of coming along with just the selection for that one trait. And so the implications of this, have been really important, and people are still doing research about how that works in our domesticated mammals today. And then Hillman's experiments with wheat showed that if you exert really strong selective pressure on wild wheat through harvesting methods, you can actually domesticate it very rapidly. So in both cases, it was a very simple experiment that showed us something really fundamental about domestication. And I've really taken a lot of inspiration from studies like these in my own work. So this is the Eastern Agricultural Complex. In this case, Eastern is referring to Eastern North America, east of the Great Plains. And these are the plants that people grew here in pre-Columbian times. So some of these crops obviously are familiar to us. Maize, beans, and squash were and continue to be extremely important to indigenous people that are from Eastern North America. But the wild progenitors of these plants are not from Eastern North America. They were first domesticated in what is now Mexico, and they reached the region where I work around 900 AD. Incidentally, it was probably a little bit earlier in New York, but that's a story for another day and quite mysterious. The plants that were domesticated in Eastern North America from native wild progenitors are other squashes like acorn squash and sunflower that we still grow today. But the other 5 plants on this slide are the lost crops. So those are the species that I'm studying and that I'm growing right now. So this is just, they're sort of in chronological order here from when they entered the crop system. So these were the earliest, followed by, they're both fall maturing pseudo cereals, so non-grass seed crops, 2 spring maturing grasses, and then erect knotweed entered the crop system last, probably around 2,500 years ago. This is the area where we have really good evidence that these plants were grown. We call it the core area because there's not a good name for it in like 21st century geography. Like it's not really the Midwest, it's not exactly the Southeast, but it made a lot of sense as a region to ancient people because of the rivers. So it's basically centered on the Mississippi River and includes most of its important northern tributaries. And then there's 2 highland areas that are on the periphery of this core area, and they've been really important because we get better preserved plant remains from caves and rock shelters. And so a lot of the evidence that I'll talk about and all of the evidence that we have that we can get ancient DNA out of comes from these peripheral highland areas. So I like to start talks about this that aren't for archaeologists by just explaining how we know that these are lost crops so that everyone doesn't spend the whole time I'm talking being skeptical. It all started with flotation in the 1960s. And one of the first things that archaeologists started to notice is they would excavate contexts like this, which is the bottom of a storage pit, the last 10 centimeters or so of which are just this dense layer of burnt plant remains. And in this case, what this turned out to be was a homogeneous mass of millions of erect knotweed seeds. Now, this is one of the assemblages that I analyzed for my dissertation. So I can also tell you that it's from a domesticated population, which is an independent line of evidence that these were cultivated plants. And I'll talk more about that in a second. But even just from the context, this is not the kind of thing that we would expect to find if people were just, you know, had these plants growing around and were occasionally using them. This is something that we would expect to find when people are storing seeds to eat them and to plant them. And then we have even more unambiguous evidence of seed storage from contexts, again, on those peripheral areas where we have dry caves and rock shelters. So maybe my favorite artifact ever is this beautiful bag from Eden's Bluff in Arkansas that's full of the seeds of domesticated chenopodium, our quinoa cousin in Eastern North America. And then this bundle of maygrass is also from Arkansas, and it looks like I could have harvested it, you know, a couple years ago, but actually it's about 2,000 years old. So we have evidence not only that people are storing these plants, but that they're storing their seeds in particular.
Natalie, do we have evidence that they were storing the seeds for replanting or for direct consumption?
Probably both, but we know they were eating them. And based on the domestication syndromes that we see, we can be fairly sure that they were also planting them. So this gets to consumption. This is a 3,000-year-old human feces, human poop. And this kind of sealed the deal, I think, for convincing archaeologists in the late 1960s and early 1970s that these were in fact crops that people were consuming. This archaeologist, Patty Jo Watson, led excavations of Salts and Mammoth Cave, which are these deep, dry caves in Kentucky. And one of the many things that they found down there were human feces. And these are, you know, have been directly dated. They're 3,000 years old, and they're full of the seeds of these plants. So hopefully, if you didn't believe that they were eating the seeds before, you're convinced now. And then sort of simultaneously, archaeologists started to send these plant remains to botanists because back then there really wasn't any such thing as paleoethnobotany. So originally, these plant remains that were coming out of archaeological sites were analyzed by botanists. And what they did is they referred back to this, you know, early literature by people like Harlan and his colleagues about domestication syndromes in annual plants to try and say, okay, is there anything about these seeds that we're getting out of the archaeological record that would indicate that they were domesticated? And we can't see all of these things in normal archaeological samples that we get, like growth habit or increase in seed set, things like that are really hard to see in the archaeological record. But there are some things that we can see, usually if we have well-preserved seeds or fruits, such as non-shattering, greater seed size, loss or reduction of germination inhibitors, and loss of glooms and other appendages that would make it difficult to process or digest seeds. So these botanists and eventually archaeologists that were trained as paleoethnobotanists started to document these changes in the archaeological record. So this is, sumpweed is the common name for Iva annua. This is from their sort of wild comparative collections. About 3 by 3 millimeters is a normal size for a fruit. And then you can see sort of from the older, these dates are a little bit wrong because this is an old paper, but from the late Archaic going up to the Mississippian, increases in seed size over time, resulting in giant seeded Iva annua by about 1,000 years ago. That's 3 to 4 times bigger than it would be in a wild population. So that's, seed size is the key criteria for identifying domesticated assemblages of Iva, which will become important later in this talk. For chenopodium, the key characteristic, although there are others, is a reduction in the thickness of its testa, which makes it germinate more easily. So this is showing, these are old species names, but basically different native phenotypes of wild chenopodium with a thick testa, like 40 to 60 microns. And then for comparison, chia, which is a domesticated chenopodium that's grown in Mexico. And then this archaeological assemblage from Russell Cave, which has even thinner testa than our modern domesticate chia down in Mexico. So again, this is for this species, that key criteria is that reduction in germination inhibitors. And then slightly less well known is little barley. Several archaeologists have argued that little barley was also domesticated either in the Southwest or in the Southeast. And the argument that they make is a little bit complex. So these are experimentally burned seeds that I burned myself this summer. So they're in like various states of undress. And you can see that after they've been burned, you still have the chaff adhering, you still have the paleolemma, the glooms, whatever, adhering to the caryopsis, the grain. And the same is true if you try to process little barley in various ways, like soaking it, grinding it, pounding it. It's really hard to get everything off of that grain. However, in the archaeological record, we only find these grains. We never find chaff, which is actually really mysterious and weird, and I don't think it's been fully explained. But a partial explanation for it is that there used to be a naked variety of little barley domesticated, like naked barley in the old world. So I'm pretty convinced by this argument, but I think that we could keep doing more research about it because it's really weird that we don't find chaff when everyone else in the world does. So this was kind of where things stood in terms of domestication research when I started doing this in 2010. So we had really clear, good, accepted evidence that sumpweed and chenopodium were both domesticated based on changes in seed size and germination inhibitors, and a pretty solid argument that little barley was as well. And then we had lots of evidence that maygrass was cultivated for a long time, even outside of its natural range, but no sort of morphological indicators that it was domesticated. And then there was erect knotweed, which was mysterious and kind of up in the air when I started doing this research. So I first heard about this mystery when I was working in the Lower Illinois Valley, which is a really beautiful region, has some of the most productive and valuable farmland in the United States today. And this is kind of looking out over the floodplain from the bluff, which is where this little site is located. So this site was somebody's house, just one family's house, about 1,000 years ago. They were farmers. And one of the things that they were growing was erect knotweed. So this assemblage of erect knotweed was really, really weird looking. First of all, it's sprouting. That's the obvious thing that's weird about it. And I don't really have time to talk about sprouts today, but if there's anyone in the audience that studies sprouts, I would love to discuss it, because I think that one way that this plant may have been processed to make it more nutritious and easier to eat, basically, would be to sprout it. So that may be intentional. But there were other things that were strange about this erect knotweed. And the botanists that analyzed it in the 1980s basically said, this doesn't look like anything in our comparative collection. It certainly doesn't look like erect knotweed. So either it's a domesticated form of erect knotweed, or else there's something missing from our comparative collection. And basically, this is how it stood for 30 years. And so when I saw this as like working on my master's, I was like, wow, like this is such a neat little question I can investigate for my dissertation. But it was actually a lot harder to answer this question than I anticipated that it would be, as it usually is. But but I ended up being able to convince myself, and then eventually, you know, a panel of anonymous botanists, that there was in fact a extinct domesticated subspecies of erect knotweed in eastern North America. And it is the type specimen for this. It comes from one of those rock shelter collections in Arkansas. So in the course of doing this research and convincing myself that this was really a domesticated population was when I first became interested in plasticity, because I was taking seed samples from you know, many different species of Polygonum first, and seeing how there was a ton of variability within and between species. And then eventually, once I'd, you know, established for myself that I was working with erect knotweed, from erect knotweed that was growing in a lot of different kinds of habitats and different places. And then eventually I was cultivating it too. And I started to notice that there's a lot of variability in what I was seeing in terms of seed morphology, depending on what was going on with the plant. So at first, I'll admit that I was just annoyed by plasticity, or I saw it as noise, and I wanted to be able to eliminate it as a possible explanation for what I was seeing in the archaeological record. But as I continued to study it longer and longer, I started to think that it could possibly be important for why this plant was domesticated, for how this plant was domesticated. So in order to explain kind of why I think that, or using this as an example, I have to explain some things about this plant, because unless there's weed scientists in the room, people probably aren't familiar with it. So it fruits and flowers all summer long. So this is a typical sort of summer plant. It has pretty much maturing fruits and flowers all on it. And then right before it dies, it does this thing where it does a big bout of simultaneous fruiting, and then the whole plant is covered in seeds, which is nice if you're a farmer and you're growing it for seed. Another important thing about this plant that you need to understand is that under normal conditions in its wild state, it produces two different kinds of fruits. So dimorphic akene is a type of fruit. It produces this one, the tubercled morph, all summer. So if you were to harvest all of the seeds off of a plant in the summer, they would all be this morph. And then it starts to produce these in the fall, but these are still adhering to the plant and they're still being produced. So any typical harvest that you take in October or November is going to be some proportion of both of these from a normal wild population. This is just like micro photographs of the thickness of the pericarp, and just to quickly show the difference. But if you had a better image, like from an SEM or something of the tubercled morph, you could see that it also has layers of wax, which keep water from getting into the seed and also mechanically restrict the germination of these seeds. So the weird thing about the domesticated assemblages that we find in the archaeological record, there's two differences. First of all, there are no tubercled morphs. They're completely missing, or at least there are very, very few of them. And also the smooth morphs are bigger than you would really expect them to be. My favorite animation, which is, which I've been using that for years, which is a classic indicator of domestication in annual seed crops. So this disappearance of the tubercled morphs is important from the perspective of farming this crop, or from the perspective of domestication, because there's a really clear germination heteromorphism between these fruit types as well, as you would expect. The smooth morphs germinate more readily. So in these experiments, these seeds were subjected to sort of standard simulated winter. So they were in moist substrate at 4 degrees Celsius for 6 weeks. And after that treatment, with some exceptions, you know, I get pretty good germination from the smooth morphs, but the tubercled morphs, that's not enough for them to germinate. And then another difference between them is that they also grow more slowly once they germinate, probably because less of the volume of the fruit is made up of that nutritive material that the seedling is using for its early growth. So this is just visualizing that in a more quantitative way. Number of leaves and height as indicators of how big the plants are at different times in the first 5 weeks of growth. And this becomes important because if you have human cultivators coming in and thinning out stands of erect knotweed at any point during the first 5 weeks of growth, they're going to remove more of the seedlings that come from those tubercled morphs. So if you have a plant in the previous generation that produced a ton of mostly smooth morphs, it's more of its offspring are going to survive this kind of an activity by people. So, okay, maybe people were thinning or maybe they weren't. Most gardeners do. Most farmers in the past that practice scatter seeding did. But in the case of erect knotweed, I think it's even more likely that people were thinning because there's a huge difference in terms of yield depending on plant density. And part of the reason why is because the plant produces seeds in the axils of its branches, so the more highly branched it is, the more seeds it produces. So in this case, you have more than twice as much seed in basically the same amount of space as these 20 plants produce. And you know, this is just showing that relationship where the less dense the plants are, the more branches they have, and then the more seeds they have. And that's interesting because in a typical wild stand, you're a very, very dense populations of erect knotweed because they drop hundreds of seeds right underneath them, right? So I don't think it's giving ancient people too much credit to suppose that they would have noticed that thinning out a stand like this could double their yield, right, in the course of a single generation. So, you know, that's a pretty neat little story of how domestication occurred. And we see this really strong, like kind of beautiful trend with only a couple of weird things going on, where you have your modern, you know, comparative sample here. And then in chronological order, you have fruits getting bigger until the very end when maybe some fertilization is going on. And then these open circles indicate the smooth morphs. So these later assemblages are almost completely composed of smooth morphs. Just ignore that. So, you know, this is a really kind of classic, like, case of domestication, what we would expect. But the problem with this neat story is that in the course of doing all of this growing erect knotweed and looking at it in the wild and stuff, I noticed some stuff that made me think that this might not be the end of the story, or it might not be the whole story. And I started to think that plasticity might be a really important part of the story. So I started to look to what people had been writing about plasticity and domestication. And of course, because I'm an anthropologist, I started with anthropology. So one of our most beloved theorists of domestication in anthropology is Mindy Zader, who works on animal domestication in the Near East. And she's been saying for, you know, it's been general, the reason why I have Putin riding the bear up here is because, you know, it's generally accepted, it has been for a long time, that some animals are more domesticable than others, right? Like there's a reason why we don't have domesticated bears, although it would be fantastic if we did. But what Mindy has been arguing for a long time is that rather than saying, okay, this animal is domesticated when we see a difference in its skeleton in the archaeological record, we should look at behavioral indicators, because there are a lot of animals that were being penned, were being bred in captivity, or ridden, or milked, or any number of really important behavioral things before we can see any morphological evidence from their bodies that they were domesticated. So I started thinking, okay, what's the equivalent to these kinds of pliability that we see in our domesticated animals in the plant world? And so I started reading what evolutionary biologists have been writing about plasticity and evolution, and realized that we're in the middle of a big to-do about it, with some biologists arguing that the modern synthesis needs to actually be revised and updated in this extended evolutionary synthesis to put more emphasis on plasticity, epigenetics, and niche construction. So this is just from one of the papers, you know, it's sort of arguing that, looking at 3 different genotypes, and they're what's called reaction norms in response to 2 different environmental types. And to give you just kind of a concrete example and a really easy to see one from erect knotweed, we've got these 2 populations, same time of year, only like an hour drive away from each other. And this one's like a little unbranched herb. And then my plants that I was growing in the garden are like giant woody shrubs. So a sort of unresolved question with this whole how important is developmental plasticity in evolution question is, how does something that's originally plastic become fixed, which is genetic assimilation? How does that become a fixed trait? And I think the mechanisms that underlie genetic assimilation, if and when it happens, are really poorly understood. And I really think that domesticates are going to be the best way to get to the bottom of this, because with domesticates, we have before and after, right? We have the wild progenitor and we have the domesticate that came from it. So we can look at traits that were originally plastic and then that have become fixed in our domesticates and try to understand how that occurred. One example, just, you know, there are other studies I could point to, but this is one that I like and I think is cool, is manioc, which in its wild state can be either a shrub or a vine, depending on whether it's growing in an open or a closed canopy. In its domesticated state, it can still be a shrub or a vine, but it's much less good at being a vine, and frequently it breaks and falls down. So it's lost some of that plasticity that it had in its wild state. So according to theorists of genetic assimilation, in order for this to occur, you need a plastic trait, obviously, something in the environment that changes permanently, because at least hypothetically, if the environment continues to be unpredictable, then it will continue to be good for the plant to be able to have these two different phenotypes, and some kind of an evolutionary cost associated with maintaining plasticity. So the non-plastic genotype is better than the plastic one. So if we take this example of the dimorphism, the fruit dimorphism, the two different types, tubercled and smooth morphs, the first question to ask is this plastic? And I discovered that it was completely by accident when my first experiment with this plant pretty much utterly failed, because I was interested in, you know, what was causing this dimorphism and blah, blah, blah. And I grew these plants in a greenhouse where they were subjected to 16 hours of light and 8 hours of darkness, no matter what season it was, and direct, you know, unlimited light right above them. And what happened was they didn't ever produce any smooth morphs. They just produced tubercled morphs, and it was a monomorphic assemblage, the opposite of the domesticated ones that we're finding in the archaeological record. So it definitely is plastic, and what the first thing I decided to investigate is what that has to do with light. So just ignore how really messy this is. My redo of this experiment this year is much nicer looking. But for a first try, it gave me some good data. And I grew the erect knotweed outside, and then I had a shady and a sunny treatment to see if it was day length, strictly day length, or like light availability in general that had something to do with the production of these two different morphs. And what I found again is that yes, it's plastic. There are significant differences between the percentage of one morph or the other that are produced by plants grown in the shade versus the sun. The shady plants produce more of the smooth morphs, but also in general, the garden grown plants produce more of the smooth morphs than any of the wild populations that I've looked at. So in particular, these are all individual plants. These three plants that produce over 90% smooth morphs, if I found those in the archaeological record, I would say that they were domesticated. Like that's outside the range of variability that I've documented in wild populations. So what is going on with these plants? I mean, they're growing in a garden, but it's not a particularly well-tended garden. And so I'm not sure what is causing this level of plasticity. And is there an evolutionary cost to maintaining plasticity? Yes, and I think I've already talked about this. If people are coming through and thinning out those plants, they're going to provide an advantage to plants that produce more of these smooth morphs, and this is just showing the same thing again. And then, are the populations subjected to new environmental conditions? So the question you have to answer first is, where would they be growing if they weren't in cultivation? And that's actually a really complicated question, because the landscape in my study area has changed so dramatically. And so it could be a whole other talk, like, well, how do we reconstruct where these plants were before the entire Midwest was taken over by kind of industrial food production? But where they grow today are in these highly disturbed, frequently flooded kind of creek banks and floodplains. So I use this as an example because it illustrates why I think, and this has been theorized by plant biologists, why this plant has two different types of fruits to begin with, right? It grows in an unpredictable environment. And sometimes what happens is, like, these plants were coming up in May, they just have their very first little flowers, and then a week later there was like a really extreme flood that scoured the riverbank. It was underwater for over a month. And when I came back there in October, there was all of that generation of plants had been wiped out. Yet the following year, the population was back. So those tubercled morphs that don't germinate very readily, they're a bet-hedging mechanism so that the population can recover after a catastrophic event, at least in theory. So again, in theory, dormancy strategies like erect knotweed and other plants that have this kind of germination heteromorphism have this strategy of escaping from unfavorable periods in time, right? And so sampling different growing seasons, whereas plants that have a very strong dispersal mechanism are sampling different locations in the same season. And these could be seen as sort of alternative ways of dealing with risk. So it stands to reason that if erect knotweed obtained a new dispersal mechanism, i.e. humans, its existing dormancy strategy would become less important for its survival. So are populations subjected to new environmental conditions? I would say, based on the archaeological record, we can say that yes, because we know that people were taking these plants up out of the floodplain and clearing locations on terraces that would otherwise be stable forest ecosystems to grow these crops. And we know that they were storing the seeds and keeping them safe, which provides an alternative to storage in the soil seed bank. So in this case, that those tubercle morphs are no longer necessary for the survival of the population because it's living in a more stable environment. And because of thinning and cultivation behavior, they're actually detrimental to the plant. So as I'm discovering all this stuff about erect knotweed and thinking, okay, maybe genetic assimilation is part of the story of how this plant was domesticated, my colleagues were coming to similar conclusions about two of the other lost crops. So my colleague Liz is working growing all of the lost crops in her garden down in Arkansas, but in particular, she was noticing some fishy business with Iva annua, sumpweed, which you can see is dwarfing her in her garden. This is not a normal height for Iva. And then my colleague Paul and his students were growing chenopodium at Ohio University and also sampling from a lot of different wild populations. So the basis of our project about plasticity and domestication rested on 3 observations that were made. Liz in her garden noticed that over the course of 4 growing seasons, the Iva started to spontaneously produce bigger seeds. This was without any kind of selection for bigger seeds. Like she wasn't thinning, she wasn't planting them deep, she wasn't doing any of the things that we've thought for generations caused the increase in seed size in our domesticated plants. And yet, if we found these seeds in the archaeological record, we would say they were kind of borderline domesticated. Like they're definitely above the cutoff for what we've considered to be domesticated Iva. So what's going on with that? I've already talked a lot about knotweed, so I'm not going to belabor it. But again, if I found these particular plants in the archaeological record, I would also say, based on the criteria that I myself just made up, that they're domesticated. So, you know, I hate to contradict myself so soon, but we may need to kind of either rethink how we're defining domestication or rethink our criteria. And finally, with chenopodium, what we're finding is that we knew that some of these seeds that have thin testas occur naturally in wild populations. And you can sort of see them with your naked eye as the ones that show up as red or yellow instead of black. They have a thinner testa. But what we're seeing harvesting from more different populations of chenopodium is that actually it can be up to, you know, like 50% of a wild plant can have these, spontaneously have these thin testas. So again, we want to figure out first of all, what's going on with chenopodium, because it's a mess just in terms of the species designations and things like that. But also what sorts of environmental conditions may be causing this variability that we see. So these 3 projects make up this larger project, the Palette for Selection, which we are doing in collaboration with Logan Kistler at the Natural History Museum, Smithsonian National Museum of Natural History. And the part of it that I'm currently executing right now is the polygonum project. So I'm redoing that messy looking experiment that I showed you from St. Louis with the sun and the shade. And we are taking RNA samples from the leaves, flowers, and developing fruits every 2 weeks so that we can try and see at what point this trigger happens where the plants start producing these smooth morphs, and if that's happening more so or earlier in one treatment versus the other, which is a good sort of way to transition into talking a little bit about the molecular facets of paleoethnobotany. So this is changing really fast. Ancient DNA, especially human ancient DNA research, has exploded exponentially in the past 5 years, and I'm hoping that we're going to see a similar kind of explosion of research in ancient plant DNA. But so far in Eastern North America, there's already been some cool stuff. It's all been based on chloroplast DNA so far, and mostly what it's been used for is biogeography. So studies of what ancient populations of squash and how they're related to modern populations of squash, and this really cool mystery about bottle gourds and how they ended up getting from Africa to North America 10,000 years ago, which is a story for another day. And then biogeography proving that, or demonstrating that chenopodium was independently domesticated in Eastern North America, and same thing for sunflowers. So molecular backup for those being from Eastern North America as opposed to some other region. And then most recently, chloroplast DNA has been used to look at sort of biodiversity in the ancient biodiversity. And so this study just came out. It's my favorite paper of 2018. And it's showing a few ancient genomes in here, or ancient plastomes, I should say, and a lot of historical and ethnographically known sunflower landraces and modern cultivars. And there's a lot of really interesting things in this paper, so I encourage you to just go read it. But one thing that I really like about it is that it's showing, first of all, that we know we've lost a lot of agrobiodiversity in the past 100 years. But even compared to that, there still are even more ancient losses that we've suffered. So one of these sort of types is not represented, one of the ancient types is not represented in any of the modern samples that we have. And also that ancient people were cultivating more than one landrace in the same place, which is cool. I mean, it's to be expected, but it's nice to have proof. So ancient epigenomics is just kind of getting started, and again, has mostly been deployed on humans, not plants, but could be really cool way to explore plasticity in plant domestication as well. So RNA has been extracted from ancient maize kernels. Methylomes have been reconstructed for ancient humans and relatives. I don't know how useful this is going to be for plant domestication, since most of our well-preserved DNA comes from seeds, and seeds are inherently not really doing much before they germinate. But hopefully, maybe we can come up with some applications for this going forward as our methods improve. And then another thing that's starting to come out of the ancient DNA research with plants is looking at kind of these deep histories of plants and their evolution in human communities. So this is sorghum. Both these studies are about sorghum. This one is kind of showing how the major land races of sorghum are correlated with ethno-linguistic groups across the African continent. So kind of looking at the connections between human cultural movement and interaction and on the one hand, and agrobiodiversity on the other hand. And this one's looking, it is a huge amount of ancient plant genomes in comparison to other studies. And what it's showing is that these really well-differentiated land races that we have in sorghum are probably pretty recent, because all of the ancient genomes are kind of falling into this undifferentiated area. And there are other conclusions in this paper that I don't have time to get into that basically indicate that sorghum, the bottleneck that where sorghum lost biodiversity with respect to its wild ancestor didn't occur at the beginning of its domestication. It occurred thousands of years later. So that's kind of contradicting what we have thought about domestication in the past. And it's highlighting the importance of, instead of looking at domestication like, okay, we've identified when this was domesticated, and then we have like when the early botanists went out and collected, and everything in between is just a wash. There's a lot of kind of evolution and different trajectories that were happening in between those 2 points that need to be investigated. So by way of wrapping up in time for some questions, I just want to just show you some of the other things that I'm doing. And as I said, I don't have like concrete results yet because we're still doing this stuff. But one of the things that we're looking at is how to get these things to germinate. It's not that easy to get wild plants to germinate in comparison to crops. And that's probably something that ancient people struggled with when they started to actually plant these. So, we started over the winter with just doing like simulated winter treatments so that we could figure out how to even get any of the seeds to germinate for our big experiments this spring. But this winter, we're going to do some other things to try and get better germination rates. And from a practical standpoint, this is important because if other people are going to pick this up and do research about it, they have to be able to get these things to germinate. It's such a headache, and it really can slow down the experiments or ruin them if it doesn't work. And then you can see like this experiment that I spent such a long time sort of setting up is right here, back here. But the majority of the area that we're cultivating is actually what I'm just calling general experiments. And what we're looking at there is just the effects of multi-cropping and plant spacing on yield and just plant health, plant size. So one of the ththings that we think based on, historically known and currently conducted Native American agriculture is that it's probably a multi-crop system. There wasn't very much monocropping going on in the New World. And so what kind of synergies there may have been between these plants in the past, we don't know how well they would do with each other versus if they're grown by themselves. And I took this picture because these are the 2 oldest lost crops, chenopodium and iva, and they grow in very similar types of environments naturally. And they seem to be doing very well together out in the field. So we'll see what kind of yields we get from them when they're in a little community versus by themselves. And then this is something that I haven't really had time to circle back to in like months because I've been out in the field all the time, but we're taking these kind of overhead shots of the plots every week so that we can hopefully get the computer to learn how to pick out the different species and calculate how much of the canopy they're making up. So I know some people here do that because I've seen posters around. So if anyone has good ideas for me about how to operationalize that, I would love to hear them. And then finally, we've noticed some kind of strange things about the lost crops, and this will just be my final kind of take. So the maygrass, which I haven't said really anything about, is called maygrass because it produces seeds in May, usually. Here it produced seeds in like mid-June because it doesn't usually grow in New York. But regardless, it produces seeds in the summer. And so some archaeologists have been wondering for years if it would be possible to get 2 crops out of Maygrass. Like if you harvested the seeds in May and then replanted them, could you get a fall crop? But it turns out that it's actually even easier than that, because if you just keep the area around the Maygrass weeded, it will die in the middle of the summer and then spontaneously regenerate in the fall. So we thought that these plants had completely senesced and were dead. And then a few weeks ago, they started sending up new seed heads, and now we're getting a second crop of Maygrass out there. So that's nice if you're a farmer. You don't have to do anything except, you know, keep it clear. And then our Iva struggled a lot. Like it really didn't do well at all. And I think the reason why is because we're way up on top of a hill on Caldwell Road. And and we had a really dry June, and it just didn't germinate well at all. So this picture is kind of harder to interpret, but this is the lowest plot. Like it's probably about 1 meter lower than the other ones, not very extreme slope. But this is the only one of these abandoned plots where nothing germinated that had any Iva, this is Iva right here, at all germinate in it. So that little difference in altitude and soil moisture, I think, although I wasn't directly measuring that, is making a huge difference in terms of whether or not this plant can germinate and grow, which is important for reconstructions of how this agricultural system worked. Because if people were trying to grow these plants together, but one of them couldn't tolerate being, you know, taken out of its sort of marshy environment, then that presents a constraint for the agricultural system. So on that note, I guess I will take questions for the next 5 minutes or so.
Thank you.
And, before I do that, I just want to especially thank my students, one of whom is here, Andrea, and Peter, for just being awesome this summer and keeping the experiments running when I've been out of town and just being really great. So thanks, Andrea. All right, anyone have questions?
Is there a salinity factor here as well? Because both chenopods and Iva are, you know, upper salt marsh plants in this part of the world.
Maybe. I mean, it's definitely a factor to be explored, although many of the places that I found them growing are not particularly salty you know, like out in the wilds of the Midwest, but it's definitely one of many factors that could be affecting how well they're doing. And because I really only had the time and resources to grow them in one location, I really wanted to do 2 different locations this summer, but it was just, I just didn't have enough manpower basically. But yeah, the soil could be a huge factor that I really want to look into in the future.
I'm assuming that the indigenous people who fed on the Chenopodium and Iva were doing so because they were nutritionally rich. Has any research been done on looking at the nutrient content of the more ancient seeds versus their contemporary varieties?
Yeah, so there has been very limited research done on the nutritional properties of these plants. There's one paper that looks at kind of like gross nutritional properties like protein versus fat versus carbohydrates. This is something that has been constrained by lack of seeds too, right? Like we want to do cooking experiments, nutritional experiments with these plants, but so far, like good farmers, we had to first see to our needs for next year in terms of planting. So one of the things that I didn't talk about, but I'm hoping to get out of this, is we're also just growing a bunch of plants in the greenhouse for seed so that we can do this kind of stuff. And it may be possible to do some kind of nutritional analyses of the ancient domesticates too, the ones that are from the desiccated collections. I just don't know enough about how that works to know for sure. I need a good collaborator.
Other questions?
Dana?
So I'm wondering in terms of why these resources fell out of cultivation and became refertilized in sort of the ancient ones, if that was related to cultural, environmental, combination of factors.
Yeah.
So my question is, so why
Actually, if she repeats it, then we'll get into Zuma.
Okay, all right. So Dana's asking basically why these crops fall out of cultivation, if it was environmental, cultural, or some kind of combination of the two. And this is a huge question that I would love to do like a long-term project on. So one of the biggest problems I think that we have with answering this question is that archaeologists really like to date the earliest occurrence of things, but they're not at all interested in dating the latest occurrence of things. So we actually don't really know when these plants fell out of cultivation because people aren't like trying to find the latest assemblage and date it. What I personally think based on, a sort of cursory literature review, is that there were at least some farmers that were still growing these crops right up until European colonization began, and that knowledge was lost along with probably lots of other knowledge as communities were moving around and people and so much warfare and destruction was happening, right? So I think that was going on, but it is undeniable that there were definitely less people growing them by the time of European colonization than had been before maize, beans, and squash arrived. So I do think that to a certain extent, once people had functioning maize landraces that worked for this climate, it was a better crop. I do think that's part of the answer, because people all over the world have adopted maize because it's highly productive. But there's probably other things going on too. In some communities, like that one where I was like, ignore this. Let me go back. This is way back. Yeah, so one of the things that's kind of like an archaeological detail has to do with that. So this assemblage here that's not domesticated, that looks like it's even smaller than the wild population, it might not even be the right species. Like these people might have been just cultivating the wrong species. I say wrong because it was different from what everybody else was ever cultivating. These are early adopters of maize. This culture is called Fort Ancient, and it's a approximately 1,000-year-old culture in the Ohio River Valley that's known for adopting maize and as a staple crop really early before other people had switched over. So it doesn't really surprise me that their knowledge of erect knotweed and their seed stock wasn't very good, considering that they were so focused on maize. So there was a lot of variability too, I guess. It's not really an answer, but somebody should write a dissertation about it someday.
Time for one more question. So can you ask Geneva if they have any questions?
We got one more in the back, and then, okay.
Domestication syndrome, one of them, of course, is shattering, and you said that's easy to determine, but how with individual seeds can you determine shattering?
So the shattering is the thing that people who study serial domestication in the old world, that's their favorite criteria because it's less like wishy-washy than seed size. And they don't actually use the seed, they use the chaff. So they'll look at the morphology of the rachis to see whether it was like a robust connection that had to be like broken, or whether it was like, I don't have an image of it right now, but it's based on experimental studies where they've looked at the morphology in wild and domesticated types. And there may be something similar for these crops. Nobody's looked at it as far as I know. But the thing is, a lot of them are into hissin in their wild state, which is also a story for another day. So, Geneva, you have any questions?
Is buckwheat old world or new world?
Sorry?
Is
Buckwheat
old world or new world?
Buckwheat is old world, and its domestication is fairly not well understood compared to a lot of other old world crops. It was probably a pretty late domesticate, and I think the latest is that it was probably domesticated on the Tibetan plateau somewhere.
Well, Natalie, I again want to thank you for a fascinating presentation. Thanks so much.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.